Good day, everyone. I'm Shane Kim Joy Laoay.
And I'm Laroga, and today we're excited to talk to you about gene therapy.
Before we discuss our presentation, let's watch a video from YouTube to better understand gene therapy.
So, what exactly is gene therapy? First, let's start with the basics. The majority of our body's genetic information is stored in 23 paired chromosomes inside the nucleus of our cells. Each chromosome is made up of DNA that stores information to determine our unique characteristics. Specific sections of DNA are called genes. As far as we know, humans have between 20,000 and 25,000 genes. Usually, a person has 2 copies of each gene, one copy passed along from each biological parent. Genes provide the instructions on how to make proteins. Proteins play an important role in how our body functions. And a small change to the DNA within our genes can alter how proteins work, which can then affect how a person breathes, walks, or digests food. To better understand how genes change, here's Dr. Hans-Peter Kiem.
Gene variants, which are genetic changes, happen to everyone and occur as cells age or are exposed to certain chemical or environmental factors. These gene variants can also be passed along from parents. Cells often recognize these genetic changes and repair them. Other times, they can cause a disease or disorder, and treatment is needed.
That's where gene therapy aims to help, by addressing the underlying cause of disease. If genes are like the blueprint to our body, gene therapy can fill in missing parts or correct errors in the drawings. Gene therapy is the use of genetic material to treat or prevent disease. The genetic material used or targeted is DNA or RNA. DNA stores our genetic information, whereas RNA is a modified copy of that information needed for the cells to build the correct protein. In gene therapy, the genetic material is transferred to the cells and then changes how proteins are produced by the cell. It can reduce levels of certain disease-causing proteins, increase production of working proteins, or even produce new or modified proteins within the cell. The genetic material needs help to get where it needs to go, like being wrapped in a package with an address label. This package delivering the genetic material is known as a vector. Typically, viruses are used as vectors because they've evolved to be very good at getting into cells. But in this case, scientists use this same ability to deliver the new therapeutic genetic material into the cell. Any disease-causing part of a virus is removed, allowing it to enter the cell without making a person sick. All vectors are tested many times for safety prior to being used in humans. There are 2 main ways to give gene therapy: in vivo and ex vivo.
In vivo gene therapy means the gene is delivered directly into the person, such as through an injection. Ex vivo gene therapy removes the person's own cells and delivers the gene to these cells outside the body. These modified cells are then returned to the person. Deciding the right approach depends on the best way to target the disease.
There are still many challenges to overcome for those seeking gene therapy as potential treatment options. One challenge is time. It's a lengthy process to test potential treatments in clinical trials and then undergo the FDA approval process. But advances in gene therapy research allow us to better understand how we could treat and hopefully someday prevent rare and debilitating diseases. For more information and resources about gene therapy and how it works, go to patienteducation.asgct.org.
So I hope you learned something from the videos, which will help you better understand our topic, which is all about gene therapy. So in this presentation, we'll explore what gene therapy is, how it works, and exciting potential for the future of medicine. So let's begin. So first, let's define gene therapy. So gene therapy is a technique that involves modifying or replacing person's genes to treat or prevent diseases. So this can be done by either inserting, altering, or removing genes within a person's cells. So gene therapy first gained attention in the late 20th century, but significant progress has been made over the past few decades. So today, it's considered a groundbreaking approach with the potential to treat many diseases that were once thought to be incurable. So why is gene therapy is so important? So gene therapy is so important as it offers hope for patients suffering from genetic disorders, cancers, and even some viral infections. So let's dive into the science behind it. So Mr. Larry Laroga will explain the basic concept of gene therapy.
So how does gene therapy actually work? So gene therapy can be broken down into two broad categories: gene editing and gene therapy. Gene editing refers to making permanent changes to the DNA, while gene therapy typically involves adding or altering genes temporarily to treat diseases. The main goal of gene therapy is to correct or replace defective genes. This can be done by inserting a healthy copy of a gene into a patient's cell, repairing a mutated gene, or replacing a gene that is not functioning correctly. In gene therapy, the target cells are typically somatic cells, which are any cells in the body except for the reproductive cells. This means the changes won't be passed down onto the future generations. Unlike germline gene therapy, which involves altering reproductive cells like sperm or egg cells, an area that remains highly controversial and regulated. Gene therapy is a treatment that aims to correct or replace a faulty gene inside a person's cell to treat genetic disorders. One of the most innovative approaches to gene therapy is stem cell gene therapy, where stem cells are used to deliver these genetic corrections. There are two main sources of stem cells used in this therapy. The first is embryonic stem cells. These are pluripotent, meaning they can become any type of cell in the body, making them highly versatile for gene therapy. The second is adult stem cells. These are multipotent and come from tissues like bone marrow or skin. They are less flexible than embryonic stem cells but are often used because they can come from the patient, reducing risk of immune rejection.
So now, let's talk about how we get those therapeutic genes into the cells. So there are two main methods of gene delivery. First is viral vectors, and the second one is non-viral methods. So viral vectors are the most commonly used delivery systems. So they take advantage of viruses, which naturally deliver genetic material to cells. Scientists modify these viruses to be harmless and insert the therapeutic gene. Examples include adenoviruses, retroviruses, and lentiviruses. So these viral vectors are highly efficient at entering cells, but the immune system can sometimes recognize and attack them, which presents a challenge in viral vectors. On the other hand, non-viral methods involve techniques like um, CRISPR-Cas9 or clustered regularly interspaced short palindromic repeats Cas9, liposomes, and electroporation. So where genes are delivered without using viruses. So while these methods tend to have fewer side effects, they can be less efficient and more difficult to control. So both approaches have their advantages and limitations, and the choice of delivery method depends on the type of disease being treated, the gene being delivered, and the target tissue. So that's all for the methods of gene delivery. Next is the type of gene therapy.
Gene therapy can be classified into two types. The first type is somatic gene therapy, and the second type is germline gene therapy. Somatic gene therapy involves modifying the genes in somatic cells, which are all cells in the body except for the reproductive cells. This is the most common form of gene therapy and the one used for diseases like cystic fibrosis or sickle cell anemia. Importantly, the changes made are not inherited by future generations. On the other hand, germline gene therapy involves modifying reproductive cells like eggs or sperm. These changes would be passed on to the offspring. Germline gene therapy holds enormous potential, but it raises serious ethical concerns and is heavily regulated as unintended consequences could affect the future generations. Currently, somatic gene therapy is the more widely used and accepted approach, and will focus on that for the rest of the presentation.
So let's proceed to the application of gene therapy. So gene therapy is being explored for a wide range of applications. So let's look at some key areas. So the first one is genetic disorders. So for diseases like cystic fibrosis, sickle cell anemia, and hemophilia, so gene therapy can potentially replace defective genes with healthy ones, so offering the chance for long-term and permanent relief. So the second one is cancer treatment. So gene therapy is also being used in cancer treatment. For instance, immunotherapy, so which involves modifying a patient's own immune system to immune cells to target cancer more effectively. So it has already shown promising results in clinical trials. So the third one is infectious diseases. So researchers are investigating ways to use gene therapy to cure viral infections like HIV and hepatitis B by altering the genetic makeup of the virus or the patient's immune system. So, so the last is the other diseases. So beyond genetic disorders, gene therapy shows promising in treating neurological diseases, eye diseases, and even muscular dystrophy. So where there are currently few treatment options. So that's all for the application of gene therapy. So the next is challenges in gene therapy.
While the potential of gene therapy is exciting, there are significant challenges that needs to be addressed. The first is ethical issues, particularly with germline gene therapy. There are concerns about designer babies, consent, and whether such technologies should be used for enhancement rather than treatment. The second concern is the safety concerns. One of the biggest issues with gene therapy is ensuring that it's safe. There are risks of immune reactions, unintended genetic changes, or even tumor formation. So, safety is a major focus in clinical trials. And last concern is technical limitations. Even though we have tools like CRISPR-Cas9 delivering genes to the right amount of cells with high accuracy, this still remains challenging. If genes are delivered to the wrong cell or the therapy doesn't last long enough, it could result in an ineffective treatment.
So despite of those challenges that Mr. Laroga mentioned, so there have been some remarkable advancement in gene therapy. So the development of CRISPR-Cas9 technology has revolutionized the field. So CRISPR allows scientists to make precise edits to DNA, which can potentially correct mutations at root cause of genetic disorders. So it's faster, cheaper, and more accurate than previous gene editing methods, opening up new possibilities for treating diseases. So additionally, we've seen successful therapy trials in treating conditions like severe combined immunodeficiency or SCID, a rare genetic disorder, and sickle cell anemia. So these breakthroughs provide a world example of gene therapy's potential to improve and even save lives. So the next is the future of gene therapy.
The future of gene therapy is incredibly promising, but it's still evolving. In the coming years, we could see a personalized medicine become more common, where therapies are tailored to an individual's unique genetic makeup. This would increase the effectiveness of the treatment and reduce the side effects. Furthermore, the scope of gene therapy could expand to include common diseases like cancer, Alzheimer's, and even diabetes that we currently don't have gene therapies for. We also expect more FDA approvals and regulatory developments as the field grows, leading to a wider availability of this treatment to the public.
So, to conclude this presentation, gene therapy represents a transformative shift in medicine. So, it holds potential to cure genetic disorders, treat cancers, and even tackle previously untreatable diseases. So, however, the challenges remain, from safety concerns to ethical issues and technical limitations. So, as research continues, we can expect even more exciting developments in this field, offering hope to millions of people worldwide. So, that's all for our presentation, and thank you for your attention.